We're here now. Okay, well, take two. Good morning, everybody. I can see that there's a fantastic room of cider technical expertise and knowledge with you at the moment, and some dialing in from elsewhere too. Thank you very much for your time, and thank you for inviting me to help host this technical and commercial cider webinar. This is an exciting opportunity to talk through some key aspects on the technical and commercial sides of cider production with, luckily for you, not just myself, or rather not really me presenting it at all, but facilitating it, but actually speaking with some absolute world-class experts. This session, this day is in two parts. This morning, we're going to have the opportunity to speak to Sigrid Gertsen Schibbye, who is the Chief Cider Technical Sales Representative on behalf of Lallemand, one of the absolute global giants and technical powerhouses for the production of enzymes, yeasts, nutrients, and bacterial cultures. And we're going to get in and have a chat with her in just a moment. But there's going to be a bit of a break where you can have some lunch and do all the things you've got to do, and then picking up a little bit later on to talk through commercial considerations for cider makers. We're going to have the opportunity to speak the man, the myth, the legend, Mr. Tom Oliver of Oliver's Cider. Some of you might have already heard of him. So we're going to get into our technical session for this morning. Before I hand over to Sigrid, who is going to be obviously very much focusing around yeast and nutrition, I'm just going to take a couple of moments to make my presentation work. First of all, there we go. And you know the nature of this presentation is you all know how to make cider very, very well. I've tasted your ciders. You are technically very good. So I'm not going to sit here and tell you how cider is made or how you made cider. But just before we get to yeast, just ensuring that we've got our bases covered in terms of understanding prior to yeast, what is the first key aspect in terms of being able to achieve the amazing array and diversity of ciders that you can, which is all part of the gloriousness of where cider is at the moment. It's about showcasing that range and those range of styles. And it starts with the selection of apple varietal, that just like with wine, the variety of the apple is all important. As you know, some of these varieties have been selected and grown for the sole purpose of making cider in some parts of Europe for centuries. There are some very old cultures with some very old varieties, whereas in other parts of Europe and around the world, cider is made with some more modern dessert and culinary apples and sometimes more sort of heritage heirloom varieties. Three key building blocks within apples are the elements which help contribute towards this amazing differentiation of different opportunities: acid, tannin, and sugar. And these have some degree of critical nature for when it comes to your selection of yeast as well, of course. First of all, when we're talking about acidity, this is malic acid. This is the natural acidity within apples. It tends to be quite an intense acidity. It's quite bold, often described as being fresh or crisp or even tart, possibly. When, from a sensorial point of view, it hits at the tip of the tongue, around the sides, provides that freshness, and it helps to contribute towards you salivating. If you ever have a very sweet cider and you don't know exactly how much acidity is in there, see how much you are dribbling. That will be able to give you an indication. And the role of acidity, other than bringing that sort of freshness, again, is helping to sort of cut through sweetness and fattiness, a really critical part, and a critical part of the majority of the ciders that you will be making in your part of the world. Tannin is a broader family of compounds which are more classically associated with the ciders of the western part of the UK and northern France, although I know for a fact that there are many people who are growing these apple varieties within the Baltic Finnish space. And these apple varieties containing tannin, that the tannin contributes towards a mouthfeel, a structure, a complexity, a mouth drying effect of astringency. It contributes potentially some taste in the form of bitterness and other flavors like earthiness, woodiness, and spiciness. And ciders made from these tannic apples will also supply some phenolic aromas. Tannins are a kind of polyphenolic compound, and the cousins of tannin's only detected in the mouth. Other polyphenolic compounds are aromatic, they're volatile, and these would be these lovely phenolic characteristics, which in their simplistic form, in their basic form, in their potentially objectively more pleasing form, would be lightly spicy, lightly woody, and earthy. But if afforded the opportunity to encounter some microbiology in the form of Brettanomyces or certain kinds of lactic acid bacteria, they can convert some of these vinyl phenolics into more ethyl phenolics, and this is where you start to get the intense medicinal, very rich, earthy, almost musty, almost manure and potentially old, sweaty horse. Everyone loves a sweaty horse first thing in the morning. And then, of course, the third building block for apples are sugar. We were just discussing very briefly there with Emmy about the sugar content and Sigrid within the fruit. That's the fuel for fermentation, and so the sugar content that's within the fruit acts as a proxy for what the potential alcohol is going to be. We've had a very hot and very dry summer here in the UK, so the sugar content of our apples is naturally going to be very high, which, as was also just mentioned, may or may not be of benefit to the cider maker, depending upon their focus and appreciation. We're primarily talking about fructose. Sorry for the typo there. Fructose with a little bit of sucrose and glucose. Very, very fermentable sugar, unlike the sugars which you would be getting within beer. Maltose and dextrose are quite complex. The yeast finds it quite difficult to convert all of those sugars to alcohol to fully attenuate. It's very easy for fructose and sucrose and glucose to ferment. And yeast, whether that be ambient wild yeasts or whether it be the introduction of a cultured yeast strain, find it pretty easy under most normal circumstances to convert all of that sugar into alcohol. There are methods by which it is possible to retain a little bit of natural residual sweetness, and if any does remain post fermentation, these sugars can contribute towards mouthfeel, texture, and enhancing of fruitiness. In the UK in 1903, the Long Ashton Research Station was the first preeminent cider research facility within the UK, and they developed this classification of apples. Initially, specifically for cider, cider apples are more tannic-rich apples in the western parts of the county, western parts of the UK, and a classification of these apples based upon their quantities of acidity and tannin. So as you can see here, a sharp apple would be considered to have a high level of acidity, and the threshold for high or low is 4.5 grams per liter of malic acid. But a low level of tannin, and that would be the threshold, there would be 2 grams per liter of tannic acid. So a sharp, high acid, low tannin. A bitter sharp, both high acid and high tannin. A bittersweet apple, low acidity and high tannin, and a sweet apple would have low quantities of acidity and low quantities of tannin. And although this was devised for initially for ciders being made within Somerset and Herefordshire and Gloucestershire over 100 years ago, this classification can and is being used all over the world for all apples which are being used for cider making, which would also include some of those specifically historically grown to make cider, but also those that have been historically grown to be for eating or for cooking or as a mixed use. And so it does provide a useful tool to understand exactly what type of apple it is that you've got. Very briefly, a word on pears. The same principles tend to apply as with apples. In areas of Europe, there are some traditional pears which have been used for making a fermented beverage. They are not generally for eating because they contain quite a lot of tannin. In other areas, perry is made with dessert and culinary pears. Two key differences to apples. Unlike apples, pears contain a proportion of sorbitol, which is an unfermentable sugar. So to a lesser, sometimes it's just a tiny bit, sometimes a considerable portion to a lesser or greater extent, these pears, these pear ciders will have some degree of natural residual sweetness. And also, these pears contain a proportion of citric acid as well as malic acid. which contributes towards the way that the taste and some of the flavor characteristics. It also does provide an easy metabolic pathway to achieving acetic acid, another volatile acidity, so it can be a little bit tricky. Okay, there we go. That's my very whistle stop tour of apples and pears to start with before we get into the importance of yeast and nutrition. Sigrid's going to be presenting. I might ask a question or two along the way. Sigrid is going to sort of pause at appropriate points once a little sort of subsection is finished. We welcome questions. I'll be looking at the chat. So if you've got any questions to put in the chat there, then I can put that to Sigrid. Obviously, we can take questions at the end. This is your opportunity to learn. I know that you're not shy. So please do bring the questions in. So Sigrid, the floor is yours. Have one
Thank you. Thank you very much. Share. Oh, wait, this one. When can you see all the pictures on the side?
No.
Oh, good. Okay. And neither the band on the top here? Okay. So you just see the full screen. Okay. Ah, fantastic. Then I can see, because sometimes I like being able to see what other people are doing. Is the sound good in the room, Anna?
Yeah.
Okay. Perfect. Excellent. Thank you very, very much for the invitation today. I'm very excited to be able to speak to you, and I wish I could be there. Maybe in another time if there's an opportunity there. You're not here to hear who I am, but just to give you a little bit of perspective, we'll focus on yeast and fermentation and apples in a moment. My name is Sigrid. I've worked with Lallemand for quite a while in different countries, but currently I'm in Denmark, so I'm sort of covering cider or apples for Lallemand in the northern area of Europe. So mostly the UK in terms of the volume, and a little bit more in Scandinavia. Unfortunately, the Baltic countries are not my area in terms of sales area, but I think in terms of as soon as anybody mentions cider, it gets pushed over to me. So I have a good chance to be able to speak to you again in the future. My background is microbiology, and I'm sure you'll be able to notice that in a few moments. I love to speak about the microbes that are involved. Working with Lallemand, I'll be speaking about some of our products, but I'm very excited if you have questions just related to general cider fermentations and microbiology. And as a company, we have great respect for spontaneous as well. That's where many of our yeast strains have come from. So it's not to say that it's a great division between introduced yeast in terms of natural yeast that we've produced versus spontaneous or natural yeast that are coming from the fermentation. So that's just a little bit of background. Just as Gabe had his apples lined up on a wall, this is some apples that I had found in our backyard when we first moved to Denmark, and it was to represent the parents and the kids. So that's my little side. I do make cider myself and have a inherent tendency towards loving cider, as it was my father is from Normandy, so it's very careful not to say from France, because Normandy was a great and powerful empire before France was even a kingdom. So he's from Normandy and Brittany, so there's a very strong link for me to love cider on many generations before. I'm hoping today to focus a little bit more on yeast and nutrition, and hopefully give you perspectives, even if you are doing a spontaneous fermentation, a little bit of this perspective of perhaps the yeast could still use a little bit of help in terms of nutrition. So that's just to give that perspective that even if you are supporting a spontaneous fermentation, when is the best time to add nutrition in its different forms? What types of nutrition are interesting in cider? Obviously, the bulk of the funding in terms of research in the world is focused on grapes, and they have a different makeup, as Gabe just pointed out, some of the key elements in an apple basis of cider, of apple acidity and sugar and acid. There's different balance in grape, but some of the relevant aspects of yeast, what yeast need of a fermentation, are still relevant. So we try and utilize some of our massive knowledge in terms of what many of my colleagues have focused on for many years in terms of studies and continuing R&D within the group, and to try and do some of our internal R&D on apple juice in the most recent years. So that, yeah, just to give you an idea, we do sometimes some case studies for different customers as they try and explore away from using perhaps the yeast that they've used for the last 20 or 30 years in a commercial operation. It's really difficult when something is working well. If it ain't broke, don't fix it. But sometimes there are cider makers who would really like to try something else, and that's risky when it's a massive tank. It's easier perhaps if it's a 20-liter or a smaller volume, depending on what's available. So in these instances, it helps a lot to do some of these pre-trials in our very controlled conditions in 1-liter fermentations where the temperature and the nutrients are added, just to give a perspective. Is it worth going this direction? Is it worth going that direction? Before scaling up to perhaps 100, 1,000, or even 50,000 liters. It depends on the scale of things. But I think it's really interesting because what we have typically seen with some of our yeast is still relevant on a cider, on an apple level, depending on the different conditions. So we can see with using perhaps say 10, 12 different yeast strains, there is an effect. One could sort of say, well, apple, the base structure of apple is very similar, so does it really have a difference? But we can see with this incidence of different aromatic components, there are some clumpings of the different yeast that look sort of either neutral in the middle, and we tend to sort of see neutral as being a negative. But if you have high quality fruit, if you have an orchard that is very sort of dominant in a certain apple type, if you want to preserve that characteristic, then neutral is not a negative. It's preserving that voice that orchard is bringing to the field. Sometimes in these cases where we're working with clients, they've sort of gone with safe and not very aromatic. And on the market that is very competitive today in terms of trying to get more aromatics, customers are looking for more freshness. Then perhaps trying to shift that a little bit is very relevant to know which yeast strains can be perhaps more floral and fruity, perhaps more floral bouquet rose versus perhaps more citrus aromas. That can be very relevant for certain clients to know. But there are still the strains that do a fantastic job that we have a lot of experience with over many years, and there's nothing negative about that. If we're saying that they are good, strong, and neutral, isn't a negative thing. And sometimes if you're looking to move perhaps from a larger operation from a smaller operation, this aspect of consistency is not a negative either, because if you have 1 year of fantastic spontaneous and the next year you have something that's very vinegary or very off, then that's not a good experience for customers either. So sometimes we can start to see customers doing perhaps a security aspect of a strain that will give you something safe every year and something that is more spontaneous and creative and perhaps a little bit more risky other years. But that it really depends on the style and the volumes that are involved. So, and spontaneous is, I think, really good as long as you are aware of it and perhaps have the experience to manage the spontaneous side of things. We'll also look a little bit about nutrition and just getting into that, just to show just one example in terms of a process. This is a juice that had, for example, some concentrate involved. And sometimes there's a lot in some of our customers, and sometimes it's just a little bit. For example, in a year that is perhaps less sugar, then adding a little bit of concentrate can help bring up to a level that is perhaps safe or consistent with what the customer is expecting in terms of versus 4% alcohol, bringing it to 6% because that's what the brand is looking for. So, but in this case, just looking at different nutrients and looking at the difference between no nutrient addition, a long and slow fermentation versus a fermentation in this case that with this client needs to be a bit more, as my husband would say, he's an engineer, optimized. Just to give you an idea, so it is Lallemand Enology. We are fruit based, and that's why I'm here today as opposed to a brewing colleague, is that we see cider as a fruit. And that's where we're sort of looking at vineyard and orchard solutions all the way through what we traditionally see Lallemand as bringing to the table of yeast. And we have been a yeast producer for over 100 years now, and that brings a depth of knowledge, but also in terms of bacteria. And not just in enology, Lallemand also does human nutrition, animal nutrition, crop security. So for example, adding organisms to a strawberry field to avoid having to spray with chemicals, but using the microbiology to protect a plant. So it's not just fermentation that we're doing for alcoholic beverages, but many others as well. And that knowledge of bacteria and how to produce bacteria for probiotics for human bacteria is used for our other organisms as well. Nutrients and protectors, as I mentioned, I'll mention a little bit here. Something that's been massive in our sort of portfolio in the last years has been the specific yeast derivative. So because we know our yeast, we also know our dead yeast. And these dead yeast can have tremendous benefits to preserving, to adding mouthfeel. So some of these amino acids, some of them we know through adding them to juices, etc. Sort of some of these amino acids are sweet and some of them are acidic, some of them are aromatic, but some of the amino acids add a volume to a product that can emulate sugar. So if you're looking to have a little bit of smoothness and aroma in sort of bringing in the acidity a little bit without just adding sugar, which is often, even in winemaking, a way to sort of quickly add mouthfeel. But if you're looking for more complexity, some of these inactive yeast can be an interesting one to look at. We also do enzymes, and chitosan I'll mention just at the end of the presentation in terms of having a preservative or an antibacterial element without adding SO2, which is extremely hot topic, along with the no and low alcohol. The other hot topic is adding less SO2, and the chitosan products can be really interesting on that level, though they don't do the full picture as SO2 can in terms of being antioxidant and antibacterial. The chitosan can make your SO2 more effective because it's taking care of the antibacterial. It allows less SO2 to be very effective in other elements of preserving a product. So it's not necessarily a no SO2, but many products aren't no SO2, it's no added SO2 because even yeast produce a little bit of SO2 sometimes. But I think what we've tried to see with using some elements, you sort of have a feeling perhaps that our products are really trying to impose sort of a commercial and industrial level, but really fermentation and yeast are quite natural, and our yeast they're a natural product as well. We've just made them in a volume and a low an element that can add a security element to a fermentation, but there is still very much an element of balance to a fermentation. And we'll look at that in terms of how we can maintain that balance as well as adding these different aspects. So we'll touch on some of these elements. I'm hoping that the next 45 minutes or whatever we have left together is still informative without being sort of going into all the details. And perhaps if there is interest to delve into some of these, then we can look at that at another time if that's possible. But if we look at nutrients to start with, nutrition is very multifaceted. I think if we look back 20 years, 30 years, we saw nutrition as a one-stop shop where you added some form of nitrogen, and then you were able to say, I've added nutrition, everything's good. But we see that as a much more interrelated, much more interesting, as with your ciders, much more complex aspect now. And it's not to make it more complicated, but it is more complex than you just sort of think of adding some form of nitrogen and then all good, we're good to go. Nutrition has an influence on the yeast growth, how quickly it grows. So if you're adding a lot of DAP at the beginning, you can overstimulate the yeast population. So adding perhaps at another timing or in a smaller dose at the beginning can be very helpful. The fermentation kinetics is useful to know if you're fermenting very cold, or if you're having, as we are right here in Denmark, having really some wonderful hot days. That has a tremendous influence as well. What we've really seen us in focusing on in the last few years are vitamins and minerals, because if you add too much of one element, then the other elements are out of balance. So apples are perhaps quite balanced. At least that's what we've been told since we're very small, that apples are the best way to keep the doctor away. But even then, there is still some elements of balance that need to be addressed if we're looking at apple fermentations. A lot of these elements of nutrition come into producing aromas. So having a more complex nutrient addition at the right timing can give you a different balance in the aromas. So adding, for example, nutrition at the very beginning tends to emphasize certain aroma production, because you're adding some of the precursors that the yeast can then use early in the fermentation. Whereas if you're adding some nutrition later in the fermentation, you're emphasizing another type of aroma production. So that in this case would be the esters. We'll touch on that in a moment, which is why we ask, how can I use all this knowledge? And that's my hope, is that in the end of this, you'll have a sort of a bigger, what's the new word? The meta view of nutrition and how you can use that in fermentations. So if we look at the fermentation, this is where my microbiologist in me comes out. I can't help but talk about lag phase and exponential phase. If we look at the beginning, whether it's a spontaneous or an addition of yeast, there is a lag phase. There is an element where the yeast that are in the juice are adapting to that juice and starting to build up. So they're slowly building. They're getting used to the pH. They're getting used to the sugar level. What is the nutrient situation? They're sort of getting a little bit of an overview, and then they're starting to build the resources and the energy to go into this exponential phase. So early on, the yeast are adapting. And then we get into this exponential phase where they're using that sugar. They're building a biomass. So they're going from one cell to 3, 4, 5, 6 cells as daughter cells. So that mother cell is growing, budding, and dividing DNA. And all that is why the yeast needs more nitrogen, because those building blocks to yeast structure are based a lot on nitrogen, as they are for us as well. When I get excited, I need a little bit of tea. And then we get to the stationary phase. So in that sort of crack where the exponential becomes the stationary phase, that's where the biomass is built up. The yeast have a way to communicate with another, and they sort of tell each other, okay, we've got enough cells to do the fermentation now. And that's really where the bulk of the fermentation will be started. So usually within that sort of 12 to 24 hours, depending on the heat, depending on the pH, depending on all those other elements in your apple must, is when there's enough biomass for the yeast to do the fermentation. And then, as in any biological system, there's a downward side at the other side, but we won't talk about that, as we often don't. Oh, shoot, there it comes. Sort of going on this idea that I work for Lallemand, I'm going to go with this idea that you are inoculating some yeast. And when we sort of have a typical recommendation of 20 to 25 grams per 100 liters, you're adding about 4 to 8 million cells, or colony forming units, CFUs. So 4 to 8 million cells per milliliter. You can see here there's a lag phase, as we just mentioned, and then this exponential phase. And then you can see in the blue the sugar starting to be utilized more actively for the fermentation. So initially, that little bit of sugar being used is to build the biomass, and then it goes into this higher activity of using the sugar that's available. If you were to under-inoculate, you can see the sort of this lower level, you can, and it can be even lower, you can see that that sort of population needs to build more, grow and divide more to come up to this sort of critical biomass that is the stationary phase. And that sort of diminishes the resources that are available for all those yeast to be able to resist any kind of shocks, temperature changes, movements, etc., that could, or lack of nutrient that could be happening. So you're diluting in a way the system, depending on your alcohol levels. For example, if you're looking to get up to, not that we want to in any cider fermentation, get up to 15% alcohol, there are customers that are doing that, but that's another type of customer than we're talking about today. But if that demand on the yeast is there to get to higher alcohol levels, anything above sort of 8 to 10 is where the yeast need to be much more resistant to ethanol toxicity and other elements that can weaken the cell at the end of the fermentation, which means that they could die before they're finished the fermentation, or they could struggle through the stationary phase. So all I wanted to illustrate was this, is that if you're under-inoculating, there can be sort of that myth or the old wives' tale that you want the yeast to work really hard. Then there are some elements that could fall apart at the end of the fermentation. So it might not be obvious right at the beginning that population is a bit weaker, but that towards the end is where you'll start to see it. And that's where it can be sometimes a problem if you want a dry cider, or you're going to re-ferment in bottle, but needed a drier cider. And here you can see where initially, if you look at the red one, you think, whoa, that fermentation is going great guns. This is a great fermentation. But reality, it's going too quickly. The fermentation, inside the cells, they can't get rid of the alcohol quickly enough. And that can cause that with the alcohol inside the cell, at the end of the fermentation, you can get a sluggish or a stuck fermentation. Whereas a little bit slower fermentation is typically easier for the yeast. They have time to develop aromas. They have time to do the fermentation properly. That can also lead to some issues if it's too slow, because if it's too cold, all the enzyme systems in the cell are slowing down. Everything is working more slowly. And that means that you can have a stuck fermentation as well. Just to give an overview, if we sort of think, okay, well, their sugar isn't perhaps a big issue for the fermentation. There are elements. We've talked about some. Temperature. If you're doing, for example, a late harvest or an ice cider, the osmotic pressure can be a tremendous stress on the yeast. So you've got much higher sugar. If you think of if you're cooking, if you enjoy cooking, if you're adding salt around something, the salt has a way of pulling the liquid out of something with that sort of gradient across a cell. For example, if you're brining a chicken before you're going to cook it, for example, there's elements of salt getting in, but you're also pulling liquid out. And that happens with you have high sugar as well. So you can be pulling liquid out of the cells, makes them much smaller. And that adds an element of stress. That means that the fermentations might not start up as quickly because the cells don't grow and divide as quickly. The other element is if you are, none of you would do this, but if you were rehydrating your yeast and you took a blender to the rehydrating yeast in those first 10 to 15 minutes when you're rehydrating, as the yeast cell is reorganizing itself from being dehydrated, it's quite sensitive. So if you were to sort of try and speed up the whole blending of the yeast in the initial stages, it has happened. You wouldn't. If you're blending it, then it can shear the cell completely apart because it hasn't been able to reestablish its cell membrane. Once it has, the cell is quite strong, but in that intermediate sort of phase of being rehydrated, it's not a good idea to blend or to really move the yeast a lot very quickly. During the fermentation, chemical, biological elements, you can see them: ethanol, starvation, not enough nutrient, the pH, these different metal ions that can come in if we are looking at which tanks we're fermenting in, and the different acid balance. It's pretty rare in a apple fermentation to have a DNA mutation, but again, it can be a stress related to all the other elements. If there's pH, if there's nutrients, those can start to come in as well. And contaminants. If you have a harvest where there's a tremendous pressure from bacteria for the apple juice, it's going to be really hard for the yeast to dominate in that instance. With all that, I just wanted to mention that there is what we call, what I call a little bit of a Superman cape that you can put on your yeast if you are going to be having an issue, whether it's colder ferments, whether it's a lack of nutrition. If there's one thing that can help your yeast be more resistant, be more viable, and those terms can be a little bit tossed around with vitality versus viability. So vital, I like to give the example of my father, who's 90 years old. He is viable. He's alive, but he's not very vital. So he's still functioning, but he's not sort of running a marathon every weekend. And that's the difference too, is you can have yeast that are alive, but the vitality at the end of the fermentation is really important. And then you can see, for example, here is where if you're adding something at the beginning of the fermentation, if you're taking care of rehydrating them carefully, if you're adding what I call this Superman cape of help to the yeast, which is the GoFirm products, the GoFirm family, and I'm going to use our products. There's other products on the market, but it's the ones I'm familiar with. So sorry about that. If you're adding these sort of Superman cape, it helps the yeast not so much at the beginning when things are good, but it really helps at the end of fermentation. And you can see this as an example, just as measuring how healthy the yeast are, how viable, how vital they are at the end of the fermentation shows that difference. So you can just see if there was a shock that came in, then the yeast is able to rebound from that a little bit more better. If you have a stuck fermentation, from experience, it's very, very rare. It's one element that has made the yeast stumble and then stop or be stuck during at the end of the fermentation. It's often a series of elements that have meant that the yeast isn't able to rebound properly. But it's not just one thing. It's not just sugar. It's not just sugar being utilized to produce alcohol. It is a very dynamic process. So just to give an illustration of all these different things going on, there is an increase in a population. There's a decrease in the sugar. There's also a decrease in the nitrogen. You can see over on the side in green, we have the nitrogen being shown. We can see the ethanol building up in light blue. You can see the glucose being utilized. It's the purple one sort of in the middle. And the CO2 being evolved. Most of our fermentations that we do are measuring the CO2. We have sort of this little, I call it the disco room, where there's lots of fermentations going on, and there's a light being turned on to generate heat so that the conditions in the fermentation are quite steady. More recently, modern developments, we have a robot that's able to do, and they can do sort of, instead of 20 or 30 fermentations that we can do, test different things. They can test, I think, 80 because of this robot being able to monitor fermentations in quite small volumes. So modern aspects coming to Lallemand as well. Yeah, the vitality of fermentation, I think I've talked a little bit about it, but just and I think we can sort of imagine where it is. It's not just at the beginning when the elements are good, there's sugar, there's nutrients. The vitality is really important to measure. And when we measure it is sort of in this growth phase and moving into the stationary phase, where the nutrients, the alcohol is beginning to increase, the nutrients are depleted, and that's where we can measure how well is the yeast able to resist this, and we can measure that with different elements of how we measure yeast vitality.
Sigrid, is it?
Yeah,
Are you able to just confirm for my benefit the difference between vitality and viability, or are they indeed the same thing?
they're not the same thing.
Okay.
They are linked for sure. So something is viable if it's functioning, it's rehydrated or it's alive. We are all viable. But the vitality is this element of how well it's not just alive, is how well it's able to resist. So it's the next level of, but they're of course linked. So
Sure.
we're measuring, if we measure, for example, the viability of the cell population, we're giving you a number. There's this many cells that are alive, there's this many cells that are dead. But we're not able, it's harder. We're working very hard to be able to give numbers, but it's sort of more an element of vitality is how well that yeast is able to spring the extra meter in the long jump. It's the how well it resists to temperature shock, pH shock, etc. It's harder to measure.
Yeah. Is this an opportunity just to take just a pause maybe and see if there
Yes.
are any
Yeah.
questions in the room, which you can either sort of come up to the screen and ask, or put something in the chat. But unless
Something you've
everybody looks
always
like this,
wondered, something
there's
that's
that there.
not clear.
Everybody looks quite happy and comfortable there, Sigs. So I would suggest carrying on.
Very good. There's just a few more, and then we'll take maybe a minute pause just to let everything sink
Gotcha.
in. What's the timing too? Yes, good. Just to give a perspective in terms of dehydrated cells, just to give, I think it's this idea that just so when you're using a package, you have a better understanding. It gives you a little bit more knowledge in terms of what you're dealing with when you have a package in your hands. When we have dehydrated yeast, there's about 8% water in this yeast that you're adding, or that you have in your hands. And that's something we've done in our process. If you're interested, I can do the sort of a tour of our production facility. There's one not too far from me in terms of where I live. So it's always interesting to see. I always get very excited to go in and see how yeast is produced. It's sort of it's impressive. It really is. So our company also makes baking yeast in Denmark, and it's a sort of iconic little yeast package. It's sort of not quite dry. It's a sort of compressed fresh yeast. It's a Maltese cross, and it's been really well distributed in Denmark. And under the pandemic, where many pictures around the pandemic of the world were toilet paper running out, in Denmark, it was the small package of 50 grams of yeast that was running out that was critical. Everybody needed to make their cinnamon swirls at home while enduring the pandemic. But there's only 8% water, so you can see when it's over on the right hand side, for example, there's a sort of shrinkage in that cell, and that's what we're trying to sort of reestablish when we're giving it those 15-20 minutes to rehydrate. If you have very cold juice, the next element is, once it's rehydrated, is to give it a few minutes of sort of time, or it's a process of adding a bit of this cold juice, so that not only is the cell adapting to the temperature, it's also adapting to the sugar levels and what are the nutrient levels. So it helps to get the yeast a bit more quickly through this lag phase. If you can adapt the yeast sort of 20 minutes, even if you have time, an hour, making sure that it gets a little bit of juice, it starts to gear up its enzyme systems and gets into a functioning cell again. So if we're looking at this sort of cell wall, that's the really important part. You can see in dehydration, it's disorganized. In rehydration, it's a little bit more, as we know it, sort of a cell membrane, this double layer of micelles that are flexible, and making sure that the inside of the yeast is healthy versus keeping the elements from the outside, such as sulfur, higher sugar levels, etc., keeping them out of the cell. And so these little elements, these sort of red elements, is what we call the sterols, or otherwise known as the survival factors. That's how important they are to the cell. And that's where we come back to, trying to tie it all together, this element of the Go Firm, which is this sort of rehydration nutrient, it's really inserting some of these extra sterols into the cell membrane, which allows a better flexibility, a better resistance of the cell membrane further down the line. If we look at what's the internal cell, it's trying to maintain its pH. If the cell at the end of the fermentation has kind of gotten a little bit leaky, sort of pitted openings, then it's really hard for that cell to maintain its internal cell pH, which allows for a healthy cell functioning. And that's where you can see sometimes the cell petering out and not being able to function properly and dying, essentially, because it's not strong enough. So it helps to take good care of your cells in the early stages in the rehydration. It just helps to, I think, emphasize how important those 15-20 minutes are of rehydration. Absolutely, you can add yeast directly to the juice. If you can see what kind of a shock it is, their cells will rehydrate, but some of them will not be vital. So some of them will rehydrate and be viable, but when it comes to the end of the fermentation, that's when you'll start to see that the vitality is not there to pull the fermentation all the way through. So relating back to Gabe's question, that's the question where you see is if you try and cut the corners too much, and some yeast strains are better than others at being sort of added directly to the juice. It depends on the juice. It depends on the temperature. So getting back to that. Other elements, the clarity. So having juice that's a little bit more clear can add a lot more aromas, can give a better fermentation. But sometimes, for example, in winemaking, you are doing flotation, you're doing clarification. So either bringing the solids to the top and getting them off from the top or adding to the bottom. Sometimes that over clear juice, there's a balance between having enough elements to have a healthy fermentation, and there's a balance between perhaps clarifying a little bit to have a better aromatic outcome. And that's where that balance is, of not having super clean juice that gives you nothing for the yeast to hang on to. You're stripping perhaps nutrients. There's no sterols for the yeast to take up and utilize, versus having a healthy fermentation. Cool. I think I'm going to jump a little bit just because I don't want to get too boring. I know there's a lot of information to come in here, but yeah, this is just an example, because what we talk about, and then I think we'll take a break after it, is Go Firm is adding sterols. When you oxygenate a juice or a fermentation, if that's possible, the addition of oxygen helps the yeast to generate some sterols or survival factors. But often in fermentation, it's anaerobic, and the aspect of pumping oxygen through your fermentation is impractical or not a sensory goal or anything of those elements. So adding oxygen could be a benefit to get the sterols in, but also that's to show that if it's not practical, the Go Firm family is able to add those sterols and avoid having a lack of sterols through the fermentation. In this case, if we were to add an oxygenation halfway through the fermentation, comparing to an addition of this rehydration protector Superman, Superwoman shield on your yeast, you can see there is still a benefit from adding these products, even if you were to add a fermentation. So it's not that adding oxygen is going to be the only way to get some oxygen to get the sterols into the cells. It can also be much more efficient to add it from the beginning of the fermentation when the yeast cells are rehydrating. But you can definitely see a difference with no oxygenation or no Go Firm added in the early stages of fermentation. Cool. I think we've covered this, and I'm going to take a minute just to let you absorb what I've been talking nonstop about. So, and then we can just jump into a little bit of nutrition.
Amazing. Thanks.
Any questions? Yeah, thank you. Is it too much? Is it helpful just to get a little bit of a pipe up? Any of you? Is it helpful to sort of just have another vision, perhaps, as you know, if you read a catalog, there's a lot of perhaps some of these elements that aren't discussed, or you don't understand why you're adding some of these products, and it can seem very commercially heavy when people like myself are promoting different products and perhaps promoting too many products. But I think sometimes for me, if you understand why, you can also adapt to your own juice and you can understand when it can be helpful versus perhaps when you aren't.
So, Asnat, is there anything in the room, any questions everyone's
Is it any
absorbing?
question? Is it too difficult, too easy? What do you know? Do you use that knowledge already, or yeah, you know?
Is it okay level?
How
Or is
do
it?
you monitor that vitality of the yeast during the fermentation process?
As I said, the vitality is really tough. I mean, we are doing, sometimes it's a question of monitoring it at one level versus another level. And this is in our labs. So it's not to say it can be done. And then you take sort of the division of those two numbers to show how the difference over a sort of a time period in the fermentation. There isn't really an easy way. You can measure the viability, and the way we do that, and many other industries use that, is with a Clini test. So it's a test for diabetes in the sugar in your blood for a diabetes test, and it's measuring the activity, how quickly the sugar is utilized in a certain 20-minute or a certain period of time. And that's what we do to measure the viability of them. So and that's something we have to do when we are guaranteeing, for example, according to OIV, the international wine rules, we have to guarantee a certain level of viability when we sell our products. And that viability has to be maintained over X amount of years. In our example, for us, it's often 4-year shelf life, where it's best before. So even after 4 years, often these products are good, but we can guarantee that it's above this minimum level for 4 years. But as with any living organism, over time, there is a decrease. So we have to make sure that within that 4 years, if it's at 95% viability when we give it to you, when we produce it, because there's always going to be some element of die-off in the drying and the packaging, etc. There's always going to be something, but over time, it'll decrease perhaps to 92, perhaps 89%, 80%, but it won't necessarily drop by 50% in 4 years. But we have to be able to maintain that if we test it 4 years down the line, that it's still above this minimum level. And I can assure you, because we've gone into this with some clients, is that often we produce to double that minimum level, just because clients expect it. They're utilizing our yeast to have an activity level that is not just minimum. Some companies will do just minimum, and that's fine. It just makes that they can perhaps produce it more cheaply, etc. But the more we understand that we have a sort of activity level that's very important for certain customers. So a great question. Unfortunately, we don't have sort of you can measure it with this quick and easy way. Nothing's quick and easy in fermentation or in life, I think. Great question. Hopefully, perhaps.
I'd like to ask about yeast. Often in the market, you can see the cider yeast. Usually it's kind of like champagne yeast or something. Do you offer some kind of, I don't know, the mixture of different bacteria? I don't know, something more interesting more than 118 or something?
Yeah, the classic example of a old but good strain and very neutral.
It's good.
Yeah, it is.
Yeah.
She is amazing, and we have another strain that's used a great deal in the UK cider industry. It's phenomenal what they can do with what's been thrown at them for the last 30 years and come out shining. So all respect to my dear yeast that are the boring choice. They've done really well. So any of our enology yeast can be used for a cider fermentation. My gut feeling is that the Saccharomyces yeast is almost too strong for a cider fermentation, a sort of a fresh juice fermentation, because Saccharomyces has been selected for wine because it gets up to 14, 16, 18% alcohol with minimal or easy fermentation capacity, and that outcompetes all these other organisms that could spoil a grape juice, a grape fermentation. The good thing is that with this interest in protection, in low SO2, etc., Lallemand has had a tremendous focus on what we call the non-Saccharomyces. And you can get me very excited about talking all these different organism names in a few minutes. But these non-Saccharomyces, I think, are going to be the way forward to finding a niche cider strain. We have tried 2 cider strains in the last few years with different collaborations with some of our research partners, both in Brittany and other places. The thing with these non-Saccharomyces, we've been producing yeast, as I mentioned, baking yeast, etc., and wine yeast for the last 3, 4, 5 decades now. They have a way of drying. They have a way. Some of them are a little bit different. Some of them, we've established that. So every strain has production notes for it to how to make it optimal. The non-Saccharomyces, it took us 5 to 10 years to master certain of these non-Saccharomyces strains. They're different. They're a different strain. They have different drying needs. They have different humidity needs. It's a great question because when I mentioned before that our yeast, when dry, are typically 8%, we went ahead with non-Saccharomyces with the same assumption because it's a yeast and we dried it. But we found that it needed between 5 and 7% humidity in the package to be able to have some kind of viability of it as a dried yeast product, non-Saccharomyces. So it took us a while to figure out what was going on. We sent very happily some of these new strains off to customers, and then 1 month later they got them and started using them during harvest, and they were dead. And until we figured out what it was, that was an issue. So we have a whole group within the Lallemand group that is optimizing strains. So when we find a new and interesting strain, they optimize the production before we bring it to the larger production because it's not always easy. So to get back to your question and to explain why there isn't 50 new and exciting cider strains, it's not as easy as it seems to bring some of these strains out. So the ones that are able to ferment a little bit, but perhaps, for example, I'll give you one example, our Biodiva, which is a Torulaspora. The Torulaspora can ferment up to 8 to 10% alcohol approximately, which is perfect for cider. It's a little bit tricky in the beginning. You have to make sure she gets started properly, but then she brings in sort of elements of cake spice, apple pie, etc., elements that are very consistent. And because she's a smaller yeast strain, a yeast species compared to the Saccharomyces, she also produces a lot of mouthfeel, which adds some elements to sort of that sort of complexity in a cider. So there are some strains that are doing well in a cider that are non-Saccharomyces that don't need to ferment up to 14% alcohol. They can go up to 8%. Also, they're much more sensitive to SO2. So if you add a little bit of SO2, these non-Saccharomyces are easier to stop than the powerhouse that is the Saccharomyces. So I hope in the coming years that we will have something that's much more fitting for cider as well. There are some liquid strains, so there are some universities that are building up sort of 1 liter liquid and selling them to the local cider fermentations because of this challenge of having to dry a strain that is a little bit more difficult. But stay tuned and keep exploring. And for sure, though, if you're just looking for something complex, more interesting than the EC1118, jump out in some of these other strains that are in a catalog, for example, and ask me or ask others what they're using. For example, in Norway, they did a trial with different yeast strains, and there was one that stood out really well for the type of apples that are being used in Norway. It was more aromatic, and that was all the other elements were the same. So the nutrition was added the same. So it wasn't just a matter of one cidery trying it and another one. It was done in a laboratory in the sort of a research institute in Norway. So there are some of these other yeasts that are really interesting, depending on which apples you have. Some of them are perhaps better if you have a lot of polyphenols, perhaps more of a red wine style yeast, because it helps to support that structure. And some of this, if you're looking for more aromatic, it could be some of these white wine strains that are really, really interesting and still secure, still good solid yeast. So it's not that you're compromising on the security of the fermentation. That was a bit long of an explanation, but yes, the answer is yes.
Yes, there we go. The short version.
That's it.
Biggs, would you be happy if your email address was shared with the participants so
Yes.
that if they had questions specific to their apples, their process, and how they might sort of explore the amazing range of different products that you've got, that they can sort of get that opportunity?
Yep, I'd be happy, and perhaps as an after slide, if I send this, I can just sort of jot some of the ones down that we have been using with a great deal of confidence in cider, in fresh apple cider fermentations. My email is my name, Sigrid, Sigrid, at Lallemand.com. So you've got that. We'll put it in the notes as well, I think, or the comments. In the interest of time, I probably will just jump into a little bit of nutrition, if that's okay. Or did you have other questions? I'm much happier to do questions, if that's...
Right topic. Maybe if we can make it shortly, if possible, but do you hear me, yeah?
Yes.
Okay, so you know that when we use wild fermentations, it's not only to get more complexity, other reason is often to slow the fermentation to the stage so that you can preserve the natural sweetness, and that after filling in bottle, it still gets a little continuation of fermentation, but still doesn't go full way. You know what I mean, yes? Understand what I'm talking about? Right.
Yes.
Then my question is there some kind of yeast strains found already, or you work on some which could do similar job, kind of convert only half of the apple sugars to alcohol and be slow enough to be safely filled in a bottles and to develop a little more pressure in a bottle, but not explode.
I think, are you talking about doing sort of a pet nat or a col fondo where you're adding?
Yeah. That's what I mean.
Yeah.
I use wild yeast, especially for this style of cider.
Yeah.
Slow the fermentation and to get stable and very natural kind of product in a bottle.
There's nothing unnatural about our
yeast. No. What I mean is that it's
okay.
non-filtered and no sulfates used for stability.
Yes. No, you're looking for a style. But what is the issue now that perhaps the yeast that you have, the wild yeast, aren't able to give enough pressure in the bottles? Is that sort of part of the element?
No, the question is, are you working on some kind of yeast which could, with wild yeast, there is a lot of unpredictability. And when we work with them, and that maybe you already developed some yeast which would be very low alcohol tolerance, maybe, and I don't know, very slow fermenting, could do the job for us easier. Similar job.
Yeah, there's several elements to that because you're looking at essentially the yeast will utilize the sugar that is available. So the amount of pressure that will be in the bottle really depends on how much sugar is left. The other element is how healthy the yeast are when they're being put in the bottle, because at that point there's very low nutrient. The increase in pressure, the CO2, can have a slowing effect. And then the temperature of the bottles when you've closed them for the last time will have an element as well. The final element is, even if the yeast have finished utilizing the sugar and they die because there's no more sugar, is what else is in that bottle that could increase the pressure even more and provide that instability of what final pressure is going to be, because if when you've closed that bottle, the yeast die and they're happy, or they're not happy, they're dead. And then you have the bacteria doing a malolactic fermentation. Even if the bacteria aren't producing a lot of CO2, they are still producing CO2, which might not fit the bottle that you've calculated, because the yeast was only supposed to add 1 atmosphere or 1.5 atmospheres. But then the CO2 from the malolactic fermentation is contributing more pressure in a bottle that might not be able to withstand that pressure. So it is a question of what is the focus of stability and what can you perhaps do. If it's a low SO2 issue, then some of these chitosan products would be extremely relevant, because it is natural. There's chitosan that comes from a shellfish, and that has to be labeled as an allergen possibility. But there are some of these products you have to check on the labels or get the spec sheet, the specification sheet. The chitosan that is made from Aspergillus, a fungus, is allowed within EU and is non-allergenic. And these chitosan products can help inhibit the bacteria that are going in. So you could add that and make sure that, because it's a matter of logarithmic how many other organisms are there. Obviously, we want the yeast to be happy and to grow to a certain critical biomass, as I mentioned before. The problem is if there's other organisms that are also building up to a critical biomass when the yeast are out of the way and not inhibiting these other organisms that are a problem. It depends on your pH as well. If you have a lower pH, the chances of having other organisms that are viable, alive and thriving are lower. But the higher the pH is, above 3.5, the more of these other organisms that can be active. So it's not an easy or quick answer. We do have some of these non-Saccharomyces yeast, but if you're doing a spontaneous, you're not looking to add other yeast. But just to give you a little snapshot, is some of these non-Saccharomyces yeast that we have that don't produce alcohol, they're being used to inhibit the wild population. So they're being added in a juice that is perhaps needs to be transported or will be chilled for a few days or a week, or the tank is going to take a day or two to fill up, and then you add these non-Saccharomyces yeast that aren't fermenting, but they're out competing some of the wild populations of bacteria, for example. Some of them actually have a direct inhibitory effect of producing a toxin that inhibits the bacteria, and other of them are just dominating because there's more of them. So I probably need to have a separate conversation with you on that. But it's if you can see what I mean, it's not as easy as just saying, yes, we have one and it does the job for you. It's a little bit more complex, because you're adding a product that's still quite dynamic in that bottle, even if it is natural and low SO2. On a microbiological level, it's probably still quite happy. So I'll chat
I think
a little bit more with
in
you.
the interests of time, Sigs, we should probably move on to the nutritional component. And we can take some more questions at the end.
Yep. Just to have an idea, is it 11:30, or are we good to go to a few minutes more? Or
I think given that we started at 10 past, we could probably go on till 22.
So we can be some minutes.
Okay, good. Okay. I wasn't sure if it was a hard stop at 10 minutes, because then I was going to fly. But thank you for the questions. They're really, and that's why I think what I'm hoping is maybe not that you go home and say, I need to add nutrition. It's more that you are able to see why, when you're adding a juice, for example, to a bottle, it's important that you have a nice and slow fermentation, whether you're adding inoculated, whether you're adding nutrition, but it's that health of the population is important, but also what's going on in the background as well, and why some of these other products are good to keep an eye on. But that might be a future presentation, is just looking at what are the other organisms, how do we inhibit those, etc. So cool. This is just to give you a little bit of an overview from Lallemand's perspective, at least. But I think in general, I think the big wide world of fermentation is how we've seen this evolution, and why in the 20s we had GoFerm, and why we now have firm sterile flash, for example, just to show we haven't just come out with a product and said, that's it, we found a solution. We very much continued to research and are finding more elements to this sort of very complex, what is yeast nutrition? So in the 1990s, when I was doing fermentations in strange parts of the world, we just looked at YAN, yeast available nitrogen. And the way to fix a low YAN was to add DAP. That's it. And sometimes you added lots of DAP all the way through the fermentation to avoid any problems, which we'll see in a moment isn't the best solution either. In the 2000s, we started looking at what is the quality of the nutrition, the nitrogen. And we could see that there was organic nitrogen versus inorganic nitrogen, and it had an effect because in the early 2000s, or in the 2000s, we came out with a product called Fermate O for Fermate Organic, mainly because the market was asking us for an organic, stamped organic product that they could add if they were an organic producer. But what we found was not only the organic producers were using it, but also the conventional fermentation were using them as well. And the feedback we were getting was really interesting. The fermentations were slower, they were more aromatic, they weren't as hot, and the yeast weren't as stressed at the end of the fermentation. So we could see that there was a definite difference in how that organic nutrient was affecting the evolution of the fermentation. And what's important there, I think, is that, yeah, we'll get into that. So then we're looking at the next level in the next sort of 10 years, and who knows what the next ones will be in the 2020s. Gosh, time's flying. How this balance of organic, inorganic, what are these other elements? What happens when you add one nutrition in the balance of things? So trying to look at the big picture of what is nutrition for the fermentation. Very quickly coming back, we're looking at the lag phase. When is the nutrition being assimilated? The nitrogen is being assimilated in the growth phase and being utilized in the stationary phase. The important element is when we're adding some of these elements to the overall look of protein synthesis, sugar transport, because nitrogen and sugar are sort of cross-related across the cell membrane. So you need enough nitrogen for the sugar to get into the cell. So there's a balance between those two that is important to remember as well. And the function of these different vitamins of oxygen, we looked a little bit before. At the end of the stationary phase, it is these survival factors that are really important to ensure that there is a membrane integrity and maintaining the cell viability, and even as well, the vitality. So if we look at these sort of, what is this big picture? We often look at these big elements, carbon, nitrogen, oxygen. We know there's lots of carbon because that's in the form of the sugar. The nitrogen is what we're looking a little bit more specifically at here. The lipids, the oxygen, there's none of that when it becomes anaerobic. The lipids we've looked about a little bit before when we talked about the cell membrane, and the vitamins and the minerals. Vitamins, minerals, they're really important in terms of enzyme cofactors. So sometimes you have an enzyme, that's this hand, and you have a cofactor, which can be a mineral that has to click into the enzyme to turn on the enzyme. Very simplistic, but just this idea of some of these vitamins and minerals are really essential to cell processes that help the cell produce a vital and a clean fermentation. So coming back to this element of balance, it is looking at vitamins, minerals, lipids, and nitrogen, which come into all of these other elements. So I'm hoping you can start to see where I'm getting at with my presentation today, and getting back to that, it is a question of balance. Ammonia, I've been using this example forever. Ammonia is a little bit like feeding a kid a sugar, lollipop, etc. It's pure sugar. Absolutely, you're going to get energy from it, but that energy is going to run out because there's nothing else other than ammonia to this sort of energy source or building block, as we see nitrogen as ammonia being. Amino acids is a little bit like giving the yeast, a child, etc., an apple, because there is sugar. It's a great example with apples. There's sugar, but there's also other elements that make that the sugar is more complex. It's taken up more slowly. There's minerals, there's vitamins, there's much more to this nitrogen. And that means that there is also an uptake order for the yeast, that they prefer some elements of this organic nitrogen versus others. But it's all coming together in terms of adding a certain element. So we see for some customers that are trying to get their yeast up to perform up to 14 or 11 or 13%, depending on the production, they need a certain amount of nitrogen. There's no two ways around it. It needs a certain number because it has to build enough population and maintain that population to a much higher alcohol. With fresh juice, I think we have that benefit of looking at what is organic nitrogen that can support a slow and steady fermentation without compromising that fermentation. So what's important to know is different yeast have different requirements. We know our yeast, we've been using them for many years, we've set them through, so we have low, medium, and high needs. So if you know you have a low nitrogen cider, then it's important to look at the yeast strains that are functioning very well, have good viability and vitality with a lower amount of nitrogen. Some of them are able to just function as humans, are able to function on less nutrients. It comes in many different levels, so we can see it in the cell metabolism. I think I'm starting to get into the sort of same points, but sometimes it helps the cell multiplication and also in terms of the cell wall integrity. A really good example here is with the Australian Wine Research Institute, where we had a control fermentation and how many days it took to finish complete the fermentation. We had, this is in grapes, sorry. In this case, there was a fermentation where they added DAP, which has been very standard sort of recommendation from the AWRI for many years, and then adding organic nutrition. If you can see the number, though, is what was really interesting, is that one of them was at 50 milligrams per liter or 50 ppm versus 24, about half of milligrams per liter, but in organic nitrogen. The difference in the days was surprising because even with half the amount of nitrogen, but organic, it was able to complete almost at the same time. This is looking at very specifics. So if we look at maybe finishing one day more or less, the organic really did hold its own. What's really interesting too is though that the sensory aspect was much more interesting on the organic because there was much more of that sort of complex nitrogen to build as building blocks for nutrients, for aromatics. Just to give you an example of a concentrate, an apple concentrate, if you look at the numbers at the bottom, so you're looking at the yan, it seems like it would be a great deal, but remembering this is a concentrate and you're diluting that apple juice back to a utilization for customers, you're looking at 25 to under 100 ppm of nitrogen in a standard apple juice. And this is irrelevant whether it's concentrate or not. We've seen that many, many times with our customers who are beginning to measure this level of nitrogen, that you're looking at under 100 in most apple juices. And it's not to say that you need to be above 100. It's just that if you're down at 20, it's a long way up for the yeast to be able to function sort of normally with that level of being quite low, below 100. For a grape fermentation, we tend to see that a minimum is 120 to 150 for a normal wine fermentation to proceed with no problems. My feeling with these last few years of doing research is that it's somewhere between 75 and 100 that leads to a healthy, happy apple cider fermentation. So that's just we obviously have to come back with that recommendation with numbers, but just looking at what we've seen these last few years. Oh, and sorry, this is where we saw the difference between this AWI project where they were looking at similar nitrogen with sort of 50 ppm versus organic on the right-hand side, and looking at these difference in terms of the aromatic complexity that came out of it. So That's just to give an example of what these different elements are. I'd just like to get into just a little bit more of the overview, I guess, in terms of ammonia, diammonia phosphate. So if you're adding DAP, an inorganic, it's not that it's bad, but it's also inhibiting the uptake of the organic nitrogen. There's a blocking of the utilization of the organic nitrogen. So if, for example, in old times, 30 years ago, you were just adding DAP and adding lots of it, the reason I gave that as an example is that you were actually blocking the uptake of what organic nitrogen there was in the juice. So you would come to the end having added this sort of inorganic nitrogen throughout the fermentation, and then you were leaving this pool of organic nitrogen in the juice that was unutilized because you'd blocked it throughout the fermentation. So this sort of more balanced approach that we've seen more recently is perhaps if it's very low, adding some DAP or a complex yeast nutrient, versus if you know that the fermentation has gone well. And that's, I think, for most people, the ability is to see, did the yeast produce some off aromas? And maybe comparing a fermentation of adding a little bit of organic nitrogen, is it much more aromatic? Is there less stress? Because sometimes a stress is at a threshold that you perhaps there's not directly sulfur components. It doesn't smell like rotten eggs, but it's not as fresh and fruity as you might have hoped. And that might be because the yeast is perhaps a bit stressed. It's not able to develop the really nice aromas, but it's not developing off aromas. So it could be interesting is to do a trial where you're adding a little bit of nutrition at the beginning and then seeing if that helps the yeast just perform that little bit better, even if it's spontaneous yeast is what I would say. So adding a little bit of nutrition when you see that the fermentation is rolling can be a really big difference for the yeast to be able to complete, not necessarily faster. That's not what I'm trying to get at in terms of efficiency, as some of the bigger customers need, is really just a clean, aromatic, happy fermentation. What can happen? I've implied it a little bit before, was these imbalances that occur. And I know we're getting into some graphs at a time in the presentation that perhaps is not, but if you stick with me a few more minutes here, this element of adding, if you have a high nitrogen, you think, oh, good, nitrogen's at the top, everything's going to go well. But what happens is there is an imbalance that happens. So you can see here with the darker, the lower graph, the darker red or the darker green, you have high nitrogen, but you have low ergosterol. So what we did was, in all of the different vitamins and minerals that we see as being critical for a fermentation, we had a high nitrogen and then a low, whichever vitamin or mineral it was. And it was so often, and sometimes drastically, and sometimes just a little bit, was that this imbalance in high nitrogen versus a low amount of either vitamins or minerals or ergosterol, and the sterols in this case, or the oleic acid, caused that the yeast was able to start functioning, but was not able to continue because of an imbalance in the high level. And what I want to be able to show you is why that happens. So I'm going to jump to this one. So if there's one that you remember, and I'm trying to perhaps sum up all the last little hour of sort of building up to population talk and fermentation chat with a yeast perspective, nitrogen is important in signaling stress. So for example, if you have a nitrogen-starved cell, you have a stress response from the cell, and it's saying, hang on, we have to buckle down. There's not really enough nitrogen here to do a really strong fermentation. So you have an adaptation that occurs in the yeast cell, and then you have a cell that doesn't go as quickly, or it doesn't produce off aromas because it's hanging on very slowly. But that means perhaps it doesn't ferment to completion either. If you have depleted for nitrogen and micronutrient starved, again, the yeast is already in that mode of protecting itself because it doesn't have enough nitrogen. So it's not going to be as critical that it's micronutrient starved because of the lack of vitamins and minerals. What happens is if you just dump in a lot of DAP or inorganic nitrogen, the yeast cells, woohoo, we've got good times, let's party. So it starts to build a population. It's got lots of DAP, but because of this lack of micronutrients, and so the vitamins and the minerals, that's not going to follow along. So there's no stress response. The yeast is going full blast, which leads to the cell vitality being impaired, and then eventually the cell death. So if we just reflect back on that other slide, it shows that why that high nitrogen level meant that the cell population actually crashed, because there wasn't that stress response that was able, the cell just sort of kept on chugging along, thinking that everything was in full swing in party mode, but there wasn't that sort of micronutrient level. And that's where these organic nitrogen elements can help, because they've got, it's a dead yeast essentially, but we've produced it, we've given it a heat treatment to release more amino acids, and that balance in amino acids and vitamins and minerals that is inherent in an organic yeast source helps to get the cell regulated properly in terms of signaling stress. I think I'm going to jump on this. This is our sort of most recent nutrient range as I was mentioning, the next swing in the 10 years of evolution is where we're looking at products that are specific to certain grape varieties or grape styles, etc. And I think the same is relevant for cider. I just need to get my colleagues to believe just as much as I do in cider being a viable group of customers to help make sure that we get the sales of Stimula cider. Oh, this one is good. Just in terms of the addition I've mentioned before, if you add nutrients early on, you have certain types of aromas that are developed. That's often, if I was to sort of categorize it in one sort of group, it's the thiols that are developed early in the fermentation. Thiols are often associated if we have sort of a Solaris or a Sauvignon Blanc type. Those are the type of aromas that you're looking for, sort of green, grassy, fresh, gooseberries, etc. And so that category is, if you're going to supplement, earlier is better. If you're trying to supplement for esters, so that sort of floral and fresh fruity aromas, an addition of a nutrient at sort of 1/3 of the sugar depletion is often the best time to sort of help the yeast swing open the doors of producing some more esters. So that gives you an idea of when we're looking at it. I think that's all I really wanted to. No, I'm not going to go through this one. You can always have it afterwards to review. Just the last one. We talked a little bit about these chitosan products, and I think it's relevant if you are a smaller facility, if you're looking to try and monitor and reduce the amount of SO2 inputs. SO2 has been used for many years. I'm sure you've all read about this and know it, but it is still a very efficient chemical when added in small amounts. And it's a shame for me that the focus in many elements, it's exciting because we're looking at what is SO2 in terms of, but if we compare the focus that wine has in terms of trying to reduce SO2 versus the focus on reducing the amount of SO2 in apricots, etc., to make them look fresh and plump, etc., the scale is massively different. So SO2 is still something that's valuable to consider, but obviously there is very much a market trend to avoid it. And trying to avoid things like rotten egg, burnt rubber, onion, garlic, and funkiness is very worthwhile to look at trying to avoid. There are sometimes elemental sulfur coming in from orchards. There's sulfur from the sulfur-containing amino acids. I didn't go into that as much today, but there are some amino acids that have a sulfur component that can be then utilized. Normally, when that happens, it's because the yeast is really going to the last of the amino acids that it really likes to use. So it's almost like a little red flag from the yeast saying, there's not a lot of nutrition left here, so I'm utilizing the amino acids that are going to give you that H2S aroma. It's the yeast way of communicating to you that there might be an imbalance or the amount of nitrogen that's available for it. Sulfur dioxide can also, and there's also sulfates occurring in apples. So it's not just the fault of sulfites being added. But what we can see with looking at some of this element is, where did it go? Sorry, I was trying to get through all the last little bits here. We can come back to the mousy taint if there's questions on that. This element of Bacillus it's a chitosan that is it's blocking the cell membranes of bacteria, and it's also bringing them out of the solution. So it's bringing them to the bottom, and it's also disrupting the cell membranes, which allows that sort of, if you were looking at how many logs, if this is logs, it's a certain million amount of bacteria per milliliter. If the Bacillus can decrease the log amount of organisms that's in your juice, it means that either your cleanliness or addition of SO2 or a racking to get off of this logarithmic decrease of your bacteria can allow that juice to be that much cleaner moving forward. So it's a question of numbers in terms of overall what is in the juice and how we can decrease that amount. It's not as effective or as wide-reaching as SO2. SO2 is, and sounds like I'm trying to promote SO2. Sorry, it is still a very effective chemical, but it's finding that balance of not utilizing or utilizing less and making sure that you still have a product that is what you want it to be when you started, when you thought, I'm going to make this product and I would like it to be fresh and clean and bright when the customer buys it in 6 months or a year. Those are the factors perhaps that we can get into in another presentation, is how do we make sure that that product is stable microbiologically with the least or the most amount of inputs, probably not the most. So that, Sigrid, I think, it gives you an idea.
Sigrid, thank you so much for giving us I suspect, a microcosm of the time, energy, and knowledge that you have the potential to contribute towards talking about microbiology and nutrition within cider. It's pretty amazing to see the world that you operate in. And for many people who come from the perspective that yeast is simply something that converts sugar into alcohol, and nutrition is simply what the yeast utilizes to make that happen. This is the gentle opening of the door. I think you've smashed down the door to showcase the incredible range and opportunity, the number of tools that cider makers can have at their disposal with the yeast selection in terms of aromatic, flavor, even mouthfeel potential that you can achieve. The critical importance of rehydration to ensure that not only is there viability, as you said, within these yeasts, and also with the nutrition, to make sure it's not about just the numbers of yeast cells, but that they're doing their job properly, not just to ensure you don't get bad characteristics, but to enhance even more kind of potential characteristics. So it's so clear to see that there's massive opportunity, and that the utilization of slightly more complex nutrition rather than just purely good old DAP can lead to again, considerable benefit. So thank you very much for your time this morning. I've put your email address in the chat there. People, if you've got questions for Sigs around yeast and nutrition, you can see the quantity and the quality of information which will come your way. So Sigrid, thank you very much. Folks, I think
Yes.